I have been interested in trying to understand how cells divide for a number of years, even before I started here. And so basically that's encoded in the DNA. Cells have a certain number of these DNA molecules, and during the cell cycle they replicate. And so a dividing cell has the task of taking these two copies and trying to equally partition them in each of the daughter cells once it divides. And so my research is focused on trying to understand the remarkable fidelity of that process. And in particular, we study the machinery that's responsible for pulling the chromosome copies apart during division. So the cell has to build this structure, which is called the mitotic spindle. And once it does this, the mitotic spindle's major function is to grab hold of these copies of the chromosomes, which are sort of together in space, linked together via a bridge. And basically this machine pulls them into each of the daughter cells during division. And so that's really the main focus of what I do. I study how that machine gets assembled. So the spindle itself is composed mostly of this polymer called a microtubule, right? And it's incredibly dynamic, but it adopts this what we call a steady state shape, which means that if you were to look at it at any given time, it looks like an American football. It looks very static. If I were to take pictures of one of these spindles, they don't do much. They're just kind of sitting there. But their individual components are turning over rapidly. So those structures, those filaments that go out and they make attachments to the chromosomes, so these cables attached to these chromosomes, and they're eventually going to end up pulling the chromosome copies apart. They're turning over with what we call a half-life of 10 seconds, which means half of them are going to be gone in 10 seconds time. The cell needs this structure to be around for probably on average, half an hour to 40 minutes, but it's turning over every 10, 20 seconds or so. So it's a pretty remarkable feat. If there's a problem in the formation of the mitotic spindle, so in this assembly process that we're interested in studying, one of the potential outcomes that can be really devastating is the complete missing of one chromosome or another, so an unequal partition of the chromosomes. And so this certainly happens with birth defects like Down syndromes, where you get an entire transfer of one chromosome or another, and it can lead to cancer. And so it's incredibly important because the division process is happening at rates that are really hard to fathom right now in our bodies. In the skin alone, there are something like 4,000 divisions per second, if you do the calculations, going on. So this is happening right now. You can't feel it, of course, but it's happening a lot, probably in the order of tens of thousands to hundreds of thousands of times in your body per second. So there are these structures that contribute to the spindles, to spindle assembly in cells, called microtubule organizing centers or centrosomes, right? And a lot of cancer cells have too many of these poles. So they go through this strange looking intermediate oftentimes that looks nothing like an American football, but instead looks kind of like a star-shaped, a three-dimensional star-shaped structure. It turns out cancer cells are really good at taking that star-shaped structure and making it look like an American football. So they take this situation where they have too many of these poles, and what they do is they focus all of those poles into one of two poles, and they get the spindle to look pretty normal. And then they can divide, and they can divide in a way where they don't have this huge mismatch of chromosome numbers in their daughter cells. One of the things that we're trying to work on in our lab is we're trying to inhibit cancer cells from going from that star-shaped spindle-like structure into something that looks normal and can function properly. And in doing so, we're going to kill the cancer cells because they won't be able to divide, or if they do, the chromosomes are going to be split up into so many daughter cells that those daughter cells won't survive. So I study it in a strange little model system. I study it in frog cells and frog egg extracts. But what we learn there certainly applies to any mammalian system, humans, other systems as well. My research program integrates sort of trying to understand the mechanics of this process, but also trying to understand the molecular mechanisms behind it, the signaling that goes on in a cell that instructs a cell to build this thing and to build it the right way. And we kind of distill our research down into two very basic questions, which are, how does the spindle get its shape, and how does the spindle get its size? We did a series of studies where we asked a very simple question, and that was, how does the size of the cell affect the size of the spindle that it can build? But that's a really tricky question to address experimentally, because for a given cell type, generally most of those cells are the same size. I can go to a different species or a different organ and find cells that are different in size, but their protein composition differs. And what we could do is we could take my extract, we could encapsulate the DNA into compartments or droplets of different sizes and ask that very question. Does the size of the droplet affect the size of the structure that we build? And it turns out it did. And the beautiful thing was, it also turned out that it followed quite nicely what's been observed in organisms. And it seems like a very trivial contribution to our knowledge, but it was really the first demonstration of that. So why is what we do important outside of trying to understand cancer? And I would say that a lot of what we do is really basic science. And a lot of what a lot of scientists do is basic science, which means that we are doing science so that we can understand how something works. We don't have an immediate new therapeutic drug or an immediate application for a lot of what we do. What we're trying to do is establish a foundation upon which other people can work to do that. You can imagine thinking about it in the opposite direction. So you can imagine a drug company wanting to develop a novel therapeutic that targets protein X, right? So why are they targeting protein X? And it's because a lot of people did a lot of research understanding what protein X does and describing it, right? And so at the time when they were doing that research, they may not have known what protein X did, or that ultimately protein X would become a target for novel therapeutic intervention, but it just so turned out it was. And I think that happens a lot, right? That basic science feeds the greater therapeutic industry, the drug industry, so on and so forth.